Thank you all very much for joining us. There's still more people coming in, but we should start. My name is Lawrence Woodward. I'm co-director of Beyond GM and A Bigger Conversation. Alongside with me this afternoon is my fellow director, Pat Thomas, and Pat will be organizing and leading the question and answer session. There is a great deal of interest in this webinar from across the board internationally, which I think highlights the issue and the concerns people have about the intersection and the interaction of far-reaching technologies with the environment, with health, with society. And we're very grateful that we've been able to attract so many participants to this webinar, because one of the things that Beyond GM and specifically our program called A Bigger Conversation has been trying to do since its inception is to try and break down the silos, break down barriers over these complex technologies, and to try and find ways to foster productive and inclusive dialogue between very different perspectives, possibly very different sets of values, to try and bridge a gap between divergent points of view over these technologies and how we as a society can use them, where they fit, where they're fit for purpose. So it's great. It's very heartening for us to see so many people from so many countries and backgrounds attending. The title we chose is Gene Editing Regulation, Acknowledging Uncertainty. We are looking at genome editing, gene editing regulation, but very much some of the same kinds of things we will be saying this afternoon, and the themes and the concerns reach out to and encompass other technologies, nanotechnology, synthetic biology, big data, and so on. This is a point at which these very emerging technologies, which are very powerful technologies, have to be given a place within society, but it has to be a place that society is happy and comfortable with. In 1984, The Guardian carried an article by a number of scientists who, in the wake of a number of ecological and environmental problems, wrote, "It is no longer feasible for ruling elites to employ experts for persuading the public that their policies are beneficial, correct, inevitable, and safe." And soon after that came the emergence of a thing that became known as post-normal science from the early 90s. And that was an approach where intrinsically there's a recognition of complexity, a recognition that a narrow view of science cannot deal with some of these complexities in a narrow scientific way. It's a recognition of the need for a greater peer group and greater transparency and more responsiveness, that science is not fixed, it changes as demands of society changes. What we have been prompted to organize this webinar with the title Acknowledging Uncertainty is that our experience here in the UK with the development of a genetic technologies bill, now an act that applies to England, is that recognition of complexity, diversity, needs for transparency, and so on, seems to have been, if not entirely thrown out of the window, but set aside. And that seems to be the emerging trend within the discussion of gene technologies in Europe as well. We are concerned about this. The acknowledgement or the failure to acknowledge uncertainty in these technologies is very concerning to us, and I think to many other people. We have to find a way to consider deregulation or reduced regulation or proportionate regulation, whichever term you care to use, in food and farming with these technologies, we have to consider them in a way that acknowledges uncertainty and in a way which acknowledges the need for transparency and acknowledges different views of risk, safety, and appropriateness. Simply, society cannot afford a failure of the science community or the regulatory community to take a very, very narrow view about specialist scientific advisors or policy experts. The nature of uncertainty and and our failure to not recognize that and get those decisions wrong is far too has far too great an impact not to get this right. We're fortunate this afternoon to have three expert presenters who have thought long and hard about these topics and about the needs and the balancing of the needs of this subject. So we're very pleased to be able to welcome these three speakers. Before I introduce our first speaker, I should just like to say that please, the chat is available. So put any comments you may make in the chat, and any questions that you want us to take up in the Q&A session, put in the Q&A section so that Pat Thomas can pick up on those and identify them. So the format will be our three speakers will make their introductory remarks, about 10 to 12 minutes. The discussion, Q&A and discussion section. As I said, this is a critical discussion, acknowledging uncertainty, how we acknowledge it, in what way we acknowledge it, and in which way we can manage it is a critical question for our time. Our first speaker is Dr. Anna Ingeborg Meyer from the Norse Research Institute in Norway. She has extensive experience in this area, a lot of thought and consideration in this area. She's an expert on biotechnology and the circular economy. She's Senior Vice President at the Independent Research Institute of Norse, and she's going to be speaking on post-normal science approach to regulation of gene-edited crops and foods.
Yes.
Anna, thank you.
Thank you, Pat and Lawrence, for this very nice introduction, and also for inviting me to give a presentation. I will start sharing my screen now, and we'll do that one. How does this look?
Very good, thank you.
Very good. Yes. I will say something about uncertainties connected to the discussions that are going on, and actually a bit about why there are so many different opinions, and maybe also a bit about what is at stake here. Because the discussions are about more than about the risks. It's more also related to uncertainty, not only related to scientific facts, but also impact on our agriculture systems, about biodiversity, and so on. But I will start to say something about regulation and the discussions about regulation. So this is a presentation of the regulatory landscape as it is of today. And actually, it's a bit wrong when it comes to Norway. I'm from Norway. The decision has just started, and this publication was from last year. As you can see here, you have New Zealand, South Africa, and Europe that is still kind of in a process. There's a lot of countries who have not started any discussions, and then there are countries that have made decisions. But all of these countries have different types of regulation, which also adds to the uncertainties about how to regulate. So it's a lot of diversity of approaches. Some countries have a GMO technology regulation, others are using the regulation they have in their countries. And there's a discussion about what should one exclude or what should one include in the new regulations. There is a lot going on definitions, and what has triggered all this is that you have this new type of genome editing techniques as CRISPR, because they have changed the time of how fast we can do animal, plant, and also breeding, and also how to use in microorganisms. It's much faster, it's cheaper, and also allows for a lot of different diverse approaches. So what are discussed are the present frameworks for regulation. Are they adequate, or should we adapt or revise these? There are different approaches and different levels. As for example, it's a discussion about, is there some of these products that should not be regulated as GMOs? And these are especially related to organisms that do not contain any novel genetic material, where you have used the CRISPR to change the genetic material in the organism you are looking at. It can, for example, be a plant, how to make it more drought tolerant. And it's a lot of different terms that are used for these techniques. It's used new plant breeding techniques, new genomic techniques, and also one are moving from calling it genetic modification to, for example, precision breeding. A lot of these processes going on are mainly focusing on plants, like the process in EU and so on. They are focusing on plants. And they who have made a decision have also made a decision on genome edited plants, that they should not be a part of the previous GMO regulations. But then you have other things happening with animals, like in Argentina and Japan. They have approved genome edited fishes, but they require that these fish should be labeled as genome edited in a given period, just to see how the market responds. And very surprisingly, it seems also that US is that a country that do not have a GMO legislation has decided to label the growth advanced transgenic salmon as bioengineered. So it's a diversity of approaches and a diversity of responses to how we are going to deal with GMOs and genome edited organisms in the regulatory landscape. And when it comes to UK and the EU and the Norway processes, which I know best, not so much about UK, but I have understood that you have just recently launched a Genetic Technology Act, which is focusing on plants. In Europe, it's a process going on. It was a decision that was made in 2018 by the European Court of Justice, that genome edited organisms should be defined under the GMO definition, and thereby be placed under current regulations, which you can see here on my slide. But after that, there's also started processes about the adequateness and how to deal with genome edited, especially plants. In Norway, just last week, we had an expert committee that was appointed by the government that published a report. I have not read all the details. It's a 500 pages report, and I will say something about it. So this gives a kind of a point to how gene technology should be regulated in Norway. And for many of you that maybe are listening, Norway is a small country, but we have an agreement with the EU, an EEA agreement, which means that we also need to harmonize our regulation with the regulations that are in Europe. So what is going on in Europe? You had the decision by the court in 2018, and then you had a report published in 2021 that was on plants, which point out that the present regulation is not fit for the purpose of some applications of genome editing and their products. and that would need to adapt this to scientific and technological processes. This was supported by some scientific reports and also some reports from the European Group on Ethics and the European Network of GMO Laboratories and so on. Very much based on a report by EFSA. So what is going on now is that one had a hearing with 70,000 input that was into the roadmap in itself about how they were going to work on the regulatory process. Then it was a hearing more detailed about including to sustainability cases and questions. And the next step will be that the Commission probably adopt this later this year. We have looked into this, and this is a kind of, we had a hearing, but that was related to the roadmap, and then it was a more kind of structured focus groups, expert groups, and so on that were included, but also possibly to give some input. But we have not seen the suggestion of the potential regulation. This has been more related to the framework and so on. And we also have seen on, if this is the first one, they are working only on plants, what about animals and also about microorganisms, which represent other issues. Anyway, this is not included in the process in Europe. So in Norway, they have looked into, they had a very broad mandate and it was just published. And they were looking both at plants, microorganisms, and also on animals in their report. They had a hearing. It was 27 reports that were submitted to this hearing. And now when it has been published, there will be a hearing process, and then it will be further work on this report by the government. So we will see what happens, and this will happen in 2024. It's also been a report in Norway who actually also discuss how genome editing and the potential implications for risk assessment by the Norwegian EFSA committee, which is called WIKM, where one of the conclusions was that actually that present EFSA guidance document on risk assessment is functional for also genome edited organisms, and they look at plants and animals and microorganisms. So a little bit about what the Norwegian report, they were not agreeing. There was a majority opinion and a minority opinion. What they agreed on was that all living organisms, plant animals and microorganisms shall be included in the regulation. That regulation should promote sustainable products, take into account consumer interests and transparency and so on. And in Norway, we had a very special regulation that has also included that there should be no risk to human health or the environment, but also that if one was going to approve GMOs, it should contribute to sustainability. It should be of some social utility and also an ethical justification for the use. And the committee, the whole committee agree on that this should be simplified, and that one should only have one additional criteria, which is ethical justifiability, that should include assessment according to utility, sustainability, fair distribution, and transparency. But then the majority group, they consider that this should only concern GMOs, because the minority group has decided that one should have a different levels of regulations, that one should have 2 for precision breeding and then 2 for GMOs. And the important issue which makes this difference is that the risk should primarily be dependent on the product's characteristics, and that the risk of a product produced with gene technology does not differ from the risk of a corresponding conventional product if the genetic changes can be considered to be similar or identical. So this means that when you're using genome editing to change a gene in an organism, that you do not bring in novel genetic material, this should be much less strict regulated and should be considered as precision breeding. while the minority group, they consider that there's no linear relationship between the technique used and the magnitude of the genetic modification, and that the potential correspondence change in the organism risk profile. So they said that even a small genotypic change can have a phenotypic or environmental consequence. So it's what made this that they didn't agree and came up with a majority and a minority opinion is the risk connected to the change you are doing by using genetic changing. But they also agree that one should change the current regulation and the current practice to better facilitate research and innovation that can contribute to sustainable products. So, but as I said, I have not read the whole document, the whole report, but anyway, there's a difference, and the difference lies in how to understand risk and uncertainty. This is an old report which have been mistaken internationally as a part of a process in Norway. This was a report that was published by the Biotechnology Advisory Board and also triggered this establishment and appointment by this National Expert Committee. And it was a very open round of hearing. It was a lot of different comments. And as you can see here, we have kind of partially gone through the comments to the hearing, and also they comment to the perception on the present Gene Technology Act. And as you can see, it's very wide, the comments. It's that the Gene Technology Act is well functioning. Others say it's too strict, it's unpredictable, it's robust, it's neutral, and it hampers competition and so on. And matters of concern for regulation is very different. It's something about that one should maintain a precautionary approach. One should restore consumer confidence. That one should change the act because it hinders sustainable development. Others point to that one should have a change towards a more product-based regulation. So there's a lot of different opinions when it comes to regulation, and also if we need to change regulation, and they are very broad. It's related to trust, it's related to democratic rights, it's related to sustainability and so on. So back to Silvio Fintovich and Jerry Ravetz, who presented post-normal science some years ago, and there's also a lot of others that have published on this. So what they point to is in that when you are in situation where actually you have uncertainties, you need to make decisions. There are a lot of different opinions. What do you deal with them? And what we can see here, there are uncertainties. There are a lot of many stakeholders. You have farmers, you have researchers. The researchers also disagree. You have policymakers, you have innovation, you have industry, and so on. So there's a lot of stakeholders and a lot of different opinions about regulation and also about the potentials. So what they point out is the need for urgency, and also that one should apply post-normal approach when it is relevant. So what about uncertainty in the debate? It's about how and what to regulate. It's related to definition of the process about using genetic modification or genome editing, and also about the definition of the product. As I said, for example, precision breeding, new genome edited techniques, and so on. It's also uncertainty related to the reasons for exclusion and inclusion of genome edited products. It's also about how to treat animals, plant, and microorganisms in these new regulations. It's related to genome editing in medical applications, and then other uses as gene drives and in synthetic biology. Its uncertainty is related to biodiversity and environment. It's genome-edited organisms can really contribute to the challenges by climate change and the loss of biodiversity, while other points out, are we sure that there are no risk to the environment and so on? and socioeconomics, who will benefit by this application and the approval and the use of genome editing, and also about contribution to sustainability. So what should we do? So what
Anna, could you begin to wrap up?
This is almost my last slide.
Thank you.
What they point to is that Jerry Ravetz and Silvia Funter, which is that we need to have much more stakeholder inclusion and engagement, and also to be a more kind of question to what the knowledge we are putting in here is a kind of who holds the knowledge, what knowledge is important, how can we validate the knowledge claims, and so on. And also about how to manage the uncertainty, what types of uncertainty addresses within the process, and what level of uncertainties are there. So the citizens' engagement is very, very important. It includes knowledge beyond the risk to health and the environment, and citizens' opinions is about science and also express values. It's not only that they lack information, but they also have information about values that are important in the society. So the goal should be actually to create a public discourse on how relevant public values should be a function as a guide for the development and regulation of genome modification and genome editing. Yeah, thank you.
Thank you, that was excellent. Thanks very much, that's great, thank you. There we are, good. So we'll press and then deal with questions together after all the three presentations. So our next presentation is from Dr. Sara Agapito, who's also from Norway. Dr. Agapito's specialities in specific area of expertise are in biochemical and genetic characterization of transgenic crops and plants and transgene flow to wild relatives, which I think is very pertinent given the differences in majority view and minority view as to how changes should be viewed and judged. So Sara, over to you.
Thank you, Lawrence. Thanks again, Pat, for the invitation, and Lawrence for organizing this, facilitating. I'm happy to share some of the research we have been doing in Norse, and also the research we are planning to do in the next few years on detection and identification. So NGT is the term that the European Commission is using for gene-edited organisms. It's called novel new genomic techniques organisms. So before I go a bit deeper into the analytical questions, I want to start this discussions with a broader view of detection. So detection is much more than just detecting one nucleotide change. There's a legal basis for this, legal basis that will establish procedures for labeling food, establishing food safety parameters, looking for unauthorized GMOs in food and in the market, surveillance processes. There's a lot of validation of these analytical methods. There are reference national labs in different countries, guidance documents that are being updated for more than a decade now. So there is a breadth of topics within detection and identification, and we should have this in mind when we discuss gene editing, and not only a very simplified yes or no, I can or I can't detect gene editing. We are running two projects in Norse now. One is called Footprint. It's funded by the Research Council of Norway. It is all about detection of gene edited products in the food chain. We have several partners which are reference laboratories in Europe, so they are aware of all these regulations, the guidance documents, and how to implement. So our research in Footprint is not only scientific curiosity, it's really towards implementing those methodologies in the EU. It includes industry, regulators, and academia, so we do have a very broad view on the topic. We have preliminary data. Unfortunately, it's not yet published, so I cannot share with you today, but hopefully soon. And we also have a SIG app project. We call it a sister project because we share samples between projects, but SIG app is more focused on molecular characterization and the usefulness of sequencing strategies for risk assessment of those products. And it's funded by the BFN, the Federal Agency for Nature Conservation in Germany. So you will hear Margaret after us. So we are also tackling the issue of these small changes, also from the risk assessment perspective. We have 2 proposals, which I can also not share much, but I really wanted to say that we are, there is a European Commission fund, the first time in all these years for detection and identification of NGT. It's a Horizon Europe project under the Farm to Fork strategy. So we have submitted a proposal there. And be surprised or not, there were only 3 proposals submitted. So that's how narrow the network and the expertise in this field is, and the reason why we need more funding to build and broader that capacity worldwide, actually. And we are also, we submitted a proposal for environmental applications of CRISPR for agriculture and also environmental purposes. So this is really new. It's a proof of concept project, something we learned from the work of the synthetic biology ATAG, which me and Margaret shared membership. So this is something we see as coming in the near future. So we wanted to anticipate and understand a bit more how to detect and identify products if they are sprayed in the field. And when we first designed the research questions of our projects, we have, of course, listed the challenges. And we were interested in scientific challenges, technical challenges, not the challenges that have been discussed around, like similarity to nature. That's not a scientific challenge. That's not what it is about. The true challenges are that we will lack the common transgenic elements within these products. The sequences that are now modified might not be unique. They might be existing in other organisms. We need very sensitivity PCR systems, and we might lack biological reference material if these products are not regulated in exporter countries, for example. So footprint deals with this true scientific questions. But there is also a typology of entity organisms. There's not only one type with one genetic modification with one nucleotide that already exists in another crop. There will be several types of gene-edited organisms. There will be organisms with small nucleotide changes. There will be organisms with large deletions, like the voxicorn. There will be insertion of new genes, new alleles. So the main point here for footprint was that we were aware that a single type of PCR method will not accommodate all this variety of organisms. We need to broaden the methodology. We need to go into different doors to detect those organisms. And that's a paradigm shift that is needed. So we had 2 main research questions. The first one was, how do the gene editing outcomes, and that's at the genetic level, how the different genetic alterations impact their detection? Is it easier to detect if we have 3 nucleotide change instead of 1, or large deletions, or a substitution in the nucleotide sequence? And what is there already? What are the new alternative detection methods that could advance our power to identify those organisms in the food chain in Europe? So the first motto for this project was like, we cannot rely on a transgenic tag. So we have to go beyond this idea that having this transgenic tag is the only way to detect gene edited. So this is a graphical abstract of our project. Of course, this was written in 2018, So we now, with our proposal to the Horizon Europe, we have even broadened and sophisticated those methodologies. But the first idea is that we cannot rely on a single PCR method. We have to explore non-PCR methods. We have to explore enhanced PCR methods. We have to test sequencing strategies and also other molecules. Are there biomarkers that could help us identify those products? And in the end, of course, to help the regulators and the industry, we would need a decision matrix. We would need some kind of tool or guidance to tell us which is the best method for this or that gene-edited organism. And we have listed the technical challenges, the analytical challenges, and how we are going to overcome that with our research projects. And the reason I added this here, I know it might sound a bit too much technical, but this is really to share with you and to speak about the real questions. To me, the detection debate has been a lot on yes or no. Oh, we cannot, like very polarized, we cannot detect. It's impossible. And then a group saying it's possible. We can always do this. You just do a PCR. And to me, it's much more complex than this. It requires refinement of the methodologies. It requires new methodologies, but that's just how science evolves and how this group, the network that has been working with this, has been doing its work for more than 10 years. So when it started with immunostrips. It started with quantitative PCR, gel-based PCR. It evolved for real-time PCR, like we could see with COVID testing. It evolved now with digital PCR, so it's even more precise and sensitive to quantify very low amounts, up to a single copy of DNA in a sample. And now we have the sequencing methodologies. So what I would like to share with you is that there are several challenges for NGT, both for approval and also for market control of unauthorized or unintentional releases. And there are ways, there are doors we can open in science for this. So just a few examples. There are no reliable detection and quantification of NGT mutations because methods are not sensitive. But there are already on the market chemistries, enhanced chemistries for PCRs, which will tackle those quantification sensitivities. And we will test with our research projects whether they can meet the minimum performance criteria as per guidelines in the European regulations. If there are no common transgenic sequences in a specific NGT, how can we screen? How can we do some surveillance? So there are sequencing methods that we can use to screen for key genetic elements, which are not the genetic elements we know of, the promoters and terminators with enhanced databases. There are no harmonized methods, validated methods. That's also a matter of working with the labs so that they could do proficiency testing of our methods. So there are several ways of overcoming this that we can try and develop. And that's now my last slide with some insights into implementation. We are aware that these methods need to be validated. We need a clear standard operating procedures so that we can lead to conclusive results and robustness for legal actions, for example, cost and time efficiency, and of course, the law enforcement authorities, they will need to be further capacitated. They will need to be trained for these new methods. But my very last piece of information to share is the Horizon Europe, the European call for research, where in their text, so this is their text of the call, they already give hints that we could look, for example, for unknown mutation in those products. We could target other parts of the genome for detection. So this is something that is being built up in the European network, and we are moving away from this dialogue of not being able to detect because it's too difficult, because there are existing sequences, etc. So we are moving in that sense with research, and we hope that in a couple of years we will be able to implement some of these methodologies in Europe. Thank you so much. I share here my email. If anyone has questions, please just forward to us. Thank you.
Thank you, Sarah. That's excellent. Raises a lot of questions and reminds me of quite a few discussions we had with researchers from mainstream in Norway and Sweden at a conference Pat and I went to, along the lines of, yes, we can do all this, but it's not necessary because we know everything. We know enough to know. So do we need to go through all of this? Which is a question maybe will come up later. And to remind participants, please feel free to post in the chat, but also post questions in the Q&A section, which is on the bottom line of the screen there. And if you can frame comments or questions in a way that we can kind of group them and make them facilitate an answer. So yeah, there's a real number of questions, and I'm sure that the next presentation, which is from Dr. Margaret Engelhardt, is going to raise even more, because we're going to talk about impacts on conservation and the wider environment. Dr. Engelhardt is the head of division for genetically modified organisms at the German Federal Agency for Nature Conservation. And it's, I have to say, I've known of this agency for a long time, and the role it's taken, not just in Germany, but within Europe, raising issues and questions about the impacts of genetic engineering technologies is really quite remarkable. And I just constantly wish that we had a similar agency asking questions in the UK. Margaret, over to you.
Yes, thank you very much. Can you hear me?
Yeah.
Right. Thanks a lot, and thanks for this invitation. I think it is important to have webinars like this, because we really have well, a complex topic, and as a risk assessor, I'm going to add another level to this discussion now. So as risk assessors, we always have to be prepared what kind of application comes and how to evaluate them, because that's our role. Currently we see some new instruments of genome editing. One has to realize that these genome editing tools can do a lot. They can do these point mutations we are currently discussing, but they can also do many point mutations. They can do transgenesis, and they also can include or change many genes that we normally refer to synthetic biology. And to our understanding, all of this is genetic engineering, especially since they all use the same tools to change this organism, and also to transform the organism themselves. Even some traditional tools are used to bring these new genes into these cells. What we also have to be prepared to, that there are really new applications coming, and also new organism groups. For example, we have decided at the Convention of Biodiversity of the United Nations, which I'm really happy about, this decision that we do a horizon scanning to help people like me to understand what kind of applications we have to be prepared for. And applications range from transgenic viruses, animals, microorganisms, trees, and also plants. Of course, we have a long tradition in agriculture plants, but even there, we already have currently on our table new applications. It's not, let's say, just the herbicide resistance and insecticides inside the transgenics, but now we have new plants that are changing the metabolism, and we as risk assessment really have to think new how to do the risk assessment. And the risk assessment also becomes more complex because the new features of these organisms are less isolated from the metabolism we have within these organisms. So we face new complexity. We also have to keep pace with these new developments to really get ready. And what I'd like to bring you today is one specific example of challenges we see coming for people like us. And these are applications that are developed for the purpose of nature conservation. So for example, you have an endangered species that is vulnerable to some illnesses, and there are discussions and ideas to change these protected species genetically, that they might have a better chance to survive. And well, being in an agency where we have the luck of having nature protection expertise and genetic engineering expertise in one house, we had a long discourse within our house, within the species protection people and the really hardcore nature protection people, and us as genetic and risk assessment expert on how to judge these applications and what could be the perspective. And it became quickly clear that the perspective is wider than the mere risk assessment, but it also touches on legal and conceptual questions that I'd briefly like to hint you to. But also we have written an expertise within our agency that maybe Pat you will put into the chat, where you can have a bit deeper look into our argumentation. So what is new about these applications? Or actually, it's in some cases, it's a paradigm shift, because some of these applications are designed to persist in nature. And so far as risk assessors, we always, especially in Europe, we always had the aim that transgenic organisms do not persist in nature, but these applications are designed for it. It is also new that nature conservation serves as a purpose and justifications for the release and dissemination of genetically modified organisms. Thirdly, it is new that these organisms are designed, or mostly new. There are only rare cases that we have releases outside agriculture's areas. For ecologists, an agriculture area is, compared to many other ecosystems, pretty simple. But now we have application in very complex ecosystems, and that will make our job more difficult. And also the last point that I already said, that maybe also protected species are genetically engineered. So from our nature conservation and perspective, how to deal with this application or these ideas that are discussed so far, they're not released, they're in the lab, it's concepts. Some of them are more developed than others, but just to make clear that they're not reality yet. But they are discussed in the international realm, in the IUCN, in the CBD. So it is good that they are discussed on this level, since we have to really think about how to deal with them. And of course, on the first level as risk assessor, we look at the mere primary risks. And of course, genome editing is a tool that helps us to change genomes at a given place. So we have at the genome or the DNA level a higher precision. However, of course, also still we have unintended genetic side effects. But what's more important, or as important, is that there might, of course, be physiological effects. So even if you have only 1 point mutation, if it is in the correct regulator, it can have numerous side effects on the physiological level. And of course, what interests us is not the degree of intervention, even though it rises. What interests us is how these organisms react in nature and interact with other organisms. So we look at the genetic level, at the metabolical level, but also on the level of interaction in nature, and or for example with non-target organisms that might feed on them and that are protected. And with this possibility of genome editing techniques with change organisms more or more complex or reach other areas within the genome that were not reachable before, we have a higher level of complexity, and with this higher level of complexity, also the uncertainty raises. And now coming back to the specific example of organisms that are meant to be released beyond the agriculture system, we add complexity because the receiving environment is more complex. And also these organisms are designed to persist in nature. So we have also another level of time and scale that we have to think about. So in our opinion, we do not see that the risk assessment instruments are ready. We are researching on them. For example, that we start to do research on modeling specific situations. But to our opinion, that the release of GMOs in the wild or in areas beyond the agriculture system, we do not think that we will be able to do risk assessment within a reasonable time, or because it is too complex to do this risk assessment. And in parallel, this is the same to predict the goals. So if you have goals that you really think that you hope to, or what these genetically changed are being done, for example, that the protected species will survive better, it will be very hard to really predict this ecologically. So to our opinion, these applications in beyond the agriculture areas, the risk assessment is not possible anymore because it is too complex. So we do not see that the instruments are ready. Of course, we are working on it, and there might be some small applications where we at the end still are able, but as long as you release it into complex receiving environment, we do not see that we have the tools at hand. So this is about the uncertainty and complexity in risk assessment. But of course, there are also, and this is, we have seen that the discourse, well, actually tomorrow I will go to a conference on science historian working on the Alizomar conference that in the very beginning of genetic engineering had discussed very widely about societal impacts. And we have seen in the last decades actually that the discourse has narrowed, at least on some levels, to the mere risk assessment. And at the CBD level, we have seen that we need to widen the discourse again and also think about further levels of the discourse, because actually all these other levels are there. The societal impacts, the socioeconomic impacts, and also the legal acts, they are in the discourse, but they're somehow at least in the risk assessment realm, they are not present anymore. So we need to see, or better see them again. So talking about these different levels, I'd like to give some little hint what kind of legal challenges might be there. So at least in many legislation, it has not been explicitly mentioned in nature conservation legislation if GMOs are part of nature conservation or not. So we can, at least, for example, we did this for the German case. We looked at the first article of this of our legislation, and with the aims of what nature conservation is heading for, and this is of course a societal decision, and to our interpretation, GMOs or changing protected species does not go along with the ideas of nature conservation, how it is put into our law, and that is also because of the ideas and the societal ideas behind why we want to protect nature. And to give you some example that are on the conceptual level, that in many societal and legislation, diversity, beauty, and utility, and nature's uniqueness is a protected good in nature conservation, and it is firmly embedded there. And if we go now and do far-reaching changes within the genetic makeup of nature, we kind of cross a border that we cross a border from the idea of protecting nature to redesigning nature, and to our understanding, this is challenging the value of nature's unique character, and therefore the Federal Agency for Nature Conservation's view, this kind of genetic engineering application would break this traditional understanding of nature with higher level protected goods and with nature conservation objectives and practices. So we do see that this from so far ideas does not go along with nature conservation. Having said this, nature conservation is a societal decision, and we deeply believe that we do need a good societal discourse on this application, because, and that is my last point, these applications are, due to their complexity, pose more uncertainty. And when you pose more uncertainty, both on the potential risks, but also the envisioned goal are also very uncertain. And in parallel, we have the situation that our biodiversity is already stressed and less resilient. So at the end, it is a societal question if we really do want to further stress biodiversity by these kind of highly uncertain applications. And I'm really happy that at the CBD level, we do think about a wider evaluation of these applications, and we are working as our agency also on tools how to better make all these different levels of the discourse visible, and for this use the expertise of technology assessment that's already around. And I think with this plea to bring this discourse into society, I'd like to end my input.
Thank you, Margaret. That's excellent. Again, excellent point. There are, Pat, I think, quite a few questions in the been posted, but just to ask the three of you to make one quick comment, really, and the difficulties of assessing and the great complexity and how that impacts. Part of that is scale. It's not just in conservation, but the more gene-edited crops appear in agriculture at greater scale in regions, it's another level of complexity. But that seems to me, and to many people I speak to, pretty clear. I mean, so the question to the three of you is, given that there is a clear case of complexity, which gets worse, gets more complex, the more scale, the more different kinds of applications and crops are around. Why is this issue not really being given enough attention by policymakers in the policy discourse that's happening in Europe? I can give you an answer as to why that's not happening in the UK, because it's been a terrible process and corrupted and so on. But can you just briefly say your view as to why this complexity, the scaling up issues are not being discussed adequately amongst policymakers in Europe? Margaret, yeah.
I could probably start. I mean, it is really, these applications sound so easy problem solver. So I do think that one level is that people want to believe that these goals are easily being achieved, and the alternative in working on the reasons for biodiversity loss, for climate change, for, I mean, in the plant section, there are, for example, currently plants being developed that are drought resistant. So these are all easy symptom healer, where you don't have to change the way of living. And I think that's why. I think that's one reason that the other way is actually the reasonable way is the hard way, is the really going to the coarse way. And it really is so tempting to believe these goals. And what we see from our agency, that of course we are the one that publish on these difficulties. We see on the risks. We publish on potential impacts on biodiversity, but it's always difficult for us to be heard, because we are the nuggers, how to say, I'm not English, but we are the one who tell the uncomfortable truths.
Can I say something?
Yeah.
Yep. I think it's also maybe relevant here is that we do not have so much experience in Europe by cultivation of GM crops. It's only in Spain that we actually do produce GM crops. And also in Europe, we have this coexistent agreement, which is kind of goes on that one should have separation, and we also have this labeling and so on, which is maybe why it's not that much discussed, and also and of course it's shown a challenge in Spain by keeping these coexistence rules, because the agriculture is small and so on. And I think this is the main reason, and also I think one challenge is that we in Europe have a different agriculture system than you have in Argentina and US and Canada, where you have big farms and produce monocrop, monocultivation. So this is what you're saying is a very important issue to take into account. Yeah. Mm.
Sarah, do you want to add anything?
Yeah, so to me, I remember having this discussion with Professor Jack Hanneman, because he wrote on scale and critical control points for risk assessment. And that to me was very timely because of the discourse on gene editing being close to nature. And the first concern that we raised was that nature does not have 7,000 hectares with the same mutation. Nature will have one or another individual in the ecosystem being vulnerable to environmental selection pressure, which is a completely different process to monitor the population of alleles in a species, which will never happen in an agricultural condition where the farmer is always adding these new seeds into the system in a large scale, like Anna was saying. So it was a very narrow narrative from whoever placed that on the debate, that those products were similar to nature in that sense. They have the same mutation. And scale will also play to be very important for environmental applications, like Margaret was explaining, something we don't, we have no experience risk assessing with classical GMOs. And gene editing is a tool that can be used in the field. So it does not require a bombardment equipment. It does not require in vitro techniques. One can really just spray CRISPR in the crop, like they are now doing with double-stranded RNA with a plane, using the same tools as you use with pesticides. So you will have genetic engineering outside the lab, but now in the field. But risk assessment does not deal with that scale issue. We do risk assessment of a single plant, regardless if it will be grown in 1 hectare or around the world. So this is really, to me, one of the most important topics that are not being properly discussed.
Pat, over to you.
Whoa, overwhelmingly busy chat and questions at the moment. There's a lot of questions around detection, which I think shows how live this issue is for people who are looking at regulation. We've got a question from Claire Robinson here, directed at you, Sarah. It says, you mentioned that detection method research can look at unintended changes in gene-edited organisms as well as intended ones for detection purposes. So will the pattern of unintended changes be consistent across all lines of a certain gene-edited GMO, for instance, the Japanese GABA tomato? Or will the changes, will the unintended changes be the same in any GABA tomato that researchers might choose to test, or will they differ from one tomato to another? In other words, how reliable is it to look at the unintended changes for detection purposes?
Yeah, that's a very recurrent question. Thank you, Claire. So we have the opportunity to clarify. So we have 2 approaches to that. One is to test the distance between an unintended change and the intended change, and the level of crossing over. What is the chance of this unintended target gets rid when crossing plants for the breeding program, for example. But the other approach is that we have to ensure that the farmer will segregate this to be present throughout the breeding program, as they do it with stacking events. So when they are stacking multiple GMOs, they have to be sure that the transgene will be present throughout the breeding process, the crossing, the back crossing. And that would have to be the same for this mark, if we chose this to be a mark. Do I advocate for that strategy to be the strategy? No, I think there will be other strategies before that one, but that is also a door we can use.
Thank you very much. The conversation around conservation has also raised several issues. One is the difference between agricultural nature and wild nature, and how they are different and why they are different, and how that affects uncertainty and risk. And there's also some questions around conservation itself, the goals of conservation itself, and whether it's legitimate to save any species that happens to be under threat, or whether we are simply interfering further with that. Margaret, would you like to have a go at that?
Yes, well, to the first part, why we think that the risk assessment is fundamentally different, even though that the risk assessment for agriculture applications is already complex and far from idle. We do see that there's really further challenges when we have this application in wild organism. For example, when you take gene drives, that this is this application where you also where you try to circumvent Mendelian rules, so that all offspring, when you release GMOs that mate with wild organism, that all offsprings are GMO at least more than 50%. There you have the challenge when you do the risk assessment, that this GMO mate with wild population where you don't know the genetic background in all cases. So you kind of get GMOs where you know some of the genetic backgrounds, but you might not know the whole background of the GMO. So this is just one example of the complexity, but of course, more challenging is the complexity that the receiving environment is more complex, and that the scale and time is more huge. But of course, we also have challenges when you take, for example, this discussion that we want to change biofilms or GM viruses, that it is very clear that we will never be able to retract them from nature. Already with agriculture organisms that are also only annual, we had seen the petunia case that they still might stay. But when you have these cases where you change many microorganisms or viruses, we don't even have a good starting idea how to do the risk assessment there. And we will not be able to do a step-by-step approach that we slowly release them and then check what happens, and if there's something risky, we get them back. So there are really new complex questions how to do a risk assessment on applications in wild populations, in microorganisms, in viruses. It's easy to probably retrieve a transgenic cow from nature, but not a transgenic virus. Coming back to these more conceptual questions, of course, that might also, well, that's a societal decision, what kind of nature we want to protect. And I remember that I gave a talk in my agency where I said, we have these people that do nature conservation for ages and all their life. And one of my colleagues stood up and said, you know, when I go in a forest and I want to rely that the tree next to me is not designed, but is grown for itself, that it is nature in a very ground sense. And so this idea of this paradigm shift from protecting nature to redesigning nature really is something that goes to, in our opinion, contradicts the nature protection we have performed so far.
Mm.
Let's put it like this.
I think the first part of that question speaks to also what was said earlier about horizon scanning, the need for horizon scanning, not just of what technologies are being used, but how they are going to be used. So for instance, there's already proposals to use gene editing in ocean-going plants and animals where control is absolutely impossible, traceability is absolutely impossible. And that really highlights the difference between agricultural nature, where there is some small element of control, and wider nature where there may be none at all. So I think it's really important to understand the difference of those two natures. There's a question for Anne. One of our audiences had to leave, but asked specifically whether Norway is the only place where societal utility and this kind of post-science approach is being practiced, or whether there are other jurisdictions where that is also being practiced. Can you say if are you alone in the universe in doing this?
Yes, no, almost. But as I've shown you, it's a bit under threat now with the new suggestions about how to deal with this. But in the EU, you have this socioeconomic inclusion into the regulation from 2015, but it's not very elaborated how to do it, and it's more kind of used as a way to say no to GMO and then pointing to specific agriculture systems and so on. So it's not this developed in Norway, where you can also argue that some risk or uncertainty can be accepted because it provides so much sustainability to sustainability and so on. So and there's also been developments under the Cartagena Protocol, which lies under the Convention of Biodiversity, where you have 172 countries who have signed it, about this import and transport of GMOs, where they also have working on socioeconomic issues. But it's not kind of what it should. It's not in the EU and not under the Cartagena Protocol been come so far to make out what does this mean. And how are we going to assess this? So it's a more, but in the process now in Europe under the Commission, there was a lift and there was an argument to include that one should assess contribution to sustainability, which of course caused a lot of interest. We got a lot of people asking about how are you dealing with this in Norway? We're considering it. So yeah.
Have there been any, has that approach led to any GMOs being approved in Norway?
Now, in Norway has been very strict. It's mostly carnations that are allowed in Norway.
Mm.
But the criteria on ethics and ethics justifiability was used to say no to a GM maize. And that was not for growing. Norway is far north and cold, but it was for import to use in feed and food of maize, and the GM maize was modified to tolerate pesticide that is not allowed to use in Norway. And then it was argued that this was a double, it was kind of not ethical justifiable to approve a product in Norway that is grown under conditions that are not allowed in Norway. Mm. Mm.
I think Margaret wants to...
May I add one little thing? I'd
Mm.
like to remind that the process in Europe is a comitology process, and that differs from the pharmaceutical sector, which is a purely risk assessment process. But the GMO process had been deliberately a comitology process. That means that there's a political level in it. And the political level, of course, also includes other arguments than the mere risk assessment arguments. And what our aim at our agency is, and also we discussed and Anne Mir also researched on it, how we can use technology assessment tools to make these other levels of the discourse more transparent in the decision within Europe. And so we are working on this kind of operationalization of technology assessment as a helping tool to make these other levels of discourse more transparent also within Europe, since it is a comitology process. And this, of course, includes also ethical, socioeconomic, and legal levels.
Thank you. I'm going to try and combine a couple of comments here. A colleague of mine who couldn't make our webinar today suggested to me that the real crisis isn't in science, it's not in regulation, it's not in innovation, but it is a crisis of academic egos that presents other views from being taken seriously. Now, we've also got a question in the Q&A about how do we bridge the gap between the technical complexities of the issues surrounding these topics and the apparent need of policymakers for simple language and frameworks. So, you know, if you could address those two questions, which I think are linked in terms of the type of advice that our policymakers get. That's to all the panel.
Annie, that's
a
for
difficult
anybody.
one.
Leap in. Margaret.
Well, one point I think is important to well, there's one level of science that is researching on these genetic engineering applications, but there's also a level of scientists that work in technology assessment, and I think it is important that we help to make these levels visible. I think they are not very often being cited by journalists. I don't know why exactly. They are not so present in these Academy of Science statements, because these Academy of Science statements are very often being done by the people who develop these organisms. And also there's a third level to it, that there isn't a huge risk assessment science community, because it is something where you cannot build academic careers on it. There's now not anymore, unfortunately, a risk assessment institute that's working on GMOs. So we also, as a Federal Agency for Nature Protection, sometimes have problem to find experts that would help us in the risk assessment research, and that's mainly because it is very difficult to build academic careers in it. There's now risk assessment research community, and that has to do with funding. That has to do also with also the power of some multinationals that do attack any publication that might be critical. We have also experienced as even federal agency this. So that's very complex, but I think that's one part of the problem.
Could I just ask Anna to clarify on that issue? The slide that you showed in Norway, where you had the majority view and a minority view, and that the slide was talking about genetic changes, or the impacts of genetic changes. On the kind of wider social utility, socioeconomic transparency questions, is that majority minority split the same, or is there a greater unanimity on the validity of those questions?
No, the whole committee agree on that one should keep it, but they want to make it more simple than it has used to be. But the majority didn't want to continue maybe to have it on the one they were kind of operating with different levels, that it should be much easier assessment on the one where you maybe have changed just a gene by using the CRISPR. So it differs between them, but it's a bit strange, and I just want to follow up with the technology assessment, because this is something that is used also in other types of technology more and more, and gives a much broader view on the use of the technology. It also points to more this case by case assessment, because as Margaret is saying, it's so different, the different ways and how you use it in plants versus bacteria and so on. I think, and also with the EU taxonomy coming in, that will play an important part. We also need to consider this kind of commercial application in a broader aspect related to sustainability and utility for whom and so on. Yeah.
Okay, we've got a couple. I know we're coming to the end here. I hope our panelists might be able to stay a few minutes on, if possible. Is that possible? Okay. Got a couple of related questions here around language and definition. One is to ask what your opinion is about the EFSA opinion that the risks of genome edited crops are similar to conventional if the change could have occurred in nature, and on the fact that EFSA has indicated not having been given a mandate to look at literature on unintended effects. This to me is related to the uncertainty around a lot of the language that genome editing is rooted in. You know, it could have, it possibly, you know, that sort of thing. And a parallel question to that is this potential inclusion of sustainability criteria, and whether this risks a kind of hierarchy of criteria, which lets politicians kind of off the hook in terms of risk assessment and shifts the power of the decision from the political to the technician. Would anybody like to dive in on either of those uncertainties?
And I maybe just to start with the EFSA opinion on mutations that could have arisen naturally. Mm-hmm. So that was a very narrow approach to how gene-edited organisms are produced. And for example, being a plant geneticist, we study a lot how mutations arise spontaneously, the frequency, and how they are maintained in a population. Because in nature, it's not about one individual, it's about the population of individuals. And that will say how the species will progress or not. Whereas in risk assessment, we were discussing this when we were talking about scale, it's about one individual and what mutation that one individual has. So EFSA approach was looking at this mutation, this single point, what they call single point mutation, one DNA base pair being changed. And that typology of mutation also exists in nature. But there are different things here. So one is the traits that these one mutation have. So to be resistant to a herbicide in nature is a mutation that it will be very unlikely to happen because there is no pressure of herbicides in nature. That's one example. Another example is what is the benefit for this mutation to be reproduced within the population and kept in this species? That's also, so there are different natural elements that happen with this specific mutation, which are not, will not happen if we grow gene-edited crops in the field. But it's also how the technology works. And that also plays with other types of mutagenic agents like radiation and chemical mutagenesis. So CRISPR works on sequence similarity, and it's not intelligent. It will always bind to whatever looks like the sequence that they have, that the complex has as a guide. And that type of mutation activity does not happen in nature. Nature does not have that typology of mutation. So plants do not have CRISPR systems. Animals do not have CRISPR systems. This is a system natural in bacteria. So the origin of these mutations is also different. So looking at the DNA sequence alone, of a bit of a DNA sequence alone from one individual, and then claiming that this process could arise in nature, to me, that's very weak. It's a weak argument. There are many other aspects that should be taken into account. And it's, I mean, if they don't have a mandate to look into literature, that's almost a joke, because how can you do a review and have a technical opinion if you cannot refer to the work of other people and learn from it?
Sarah, how much of what you've just said also applies to so-called traditional or conventional breeding? So in the UK Act, they talked about nature, and then actually in the Act, they dropped mention to nature and are now majoring on so-called traditional breeding. I think the term in Europe is being used as conventional breeding. So how much of what you've just said in regards to CRISPR applies to traditional or conventional breeding?
Yeah, so if we take the concept of traditional breeding being crossing of species that are sexually compatible, that type of generating diversity, because it's when you cross individuals, you're generating diversity. If you respect sexual barriers, you will also respect the way the chromosomes interact, the linkage of genes, and you won't disrupt that. If you do protoplast fusion, you know, more when you go over sexual barriers, then you might go over some of these natural systems to maintain the integrity of the chromosomes and the genes and how they are connected and how they interact. A very good example for me is the waxycorn, because I had a chance to read the waxycorn dossiers from Curtiva, and there they made a very good statement saying the allele that it has been silenced in the waxycorn already exists in nature. It's not the same sequence, but the allele functions the same. However, we did not use that allele because it's linked to several other genes, like we call quantitative trait lossy. It's several other genes that would affect our yield. We could not work this doing conventional crossing. Therefore, we did gene editing. So when you do gene editing in that sense, you are breaking that organization, that structure in the genome, because you couldn't have done this with conventional breeding. But that's a case-by-case risk. I'm not saying that that is a risk because you're doing this. But there are different routes. So the analogy of saying it's the same, from my perspective, it's not, and that should be investigated. Whatever.
Yeah, it's a massive piece of disingenuity on the part of the conventional scientific community, saying that these things could have been achieved by traditional breeding. Only if you include destruction of cell walls and protoplast fusion within the definition of traditional breeding.
Yeah, there is confusion also with marker-assisted selection, for example, when you use PCR just to track genes along your breeding program. That does not interfere at all. You're just tracking, like if you're doing detection all over. But there is also confusion that this might be some kind of genetic manipulation.
It really it challenges this notion, what you're saying, of so-called precision breeding. When you talk about CRISPR not being intelligent, and of course what we hear also is in terms of the mutagenesis question is, you know, we've been doing it for years, it's perfectly safe, there's no problem with it. It doesn't need to be regulated, and in some ways it's more damaging than so-called precision breeding. So why should we regulate precision breeding? And this is a question that's come up from one of our audience, is how do you respond to that?
So I'd like first to also respond to the first question of the naturality and breeding argument, because I'm again have the risk assessor perspective, and the reason why we do GMO law is that we pose the question if these organisms have risks or not. And for us, it's pretty missing the point if we discuss if it's similar to nature or not. In nature we have, and also in traditional, well, mainly in nature, we also have, for example, transgenesis. And even though that in nature we have transgenesis, we decided to have a GMO law. And that's why it might pose risks. And I sometimes find it interesting why this question is not the key question. It's fully irrelevant. If it's similar to nature or breeding, it is fully irrelevant. The only thing that's relevant if it poses risks or not. And if it poses risks, and if we do it deliberately, we are responsible. Let me give you an example. If you have a point mutation, a virus, that an animal virus becomes pathogenic to humans. And if it happens in nature, we cannot prevent it. Okay, that's like this. But if we do it deliberately, we are responsible for it, and we have to prevent that these harms would be done. So that's the question. So the whole breeding, or if it's natural, we have it in gene drive, we have the discussion, we also have it in nature, it's irrelevant. The only question if it poses risks or not.
Anyone else? Anne, you unmuted yourself, yeah?
Yes, I just want to follow up with Margaret. The purpose by a regulation is to protect environment and also to protect human health. It's a kind of to be sure that the food we are eating is safe, that the environment are not affected, and so on. So sometimes these themes are forgotten, and then some of the argument is of course linked to the risk, that there are less risk, there are more precision, but also that the regulation is time consuming, it costs a lot of money, it hampers competition and so on. So it's a lot of arguments on both sides, which have different origins and so on. So it's an interesting debate, and we are in a time of climate crisis. We have a biodiversity crisis. We have a transformation of society that is going on, at least especially for us that are dependent on fossil fuel. We have the war in Ukraine, which is really, it's kind of where are us as a society moving, and then to make these changes and so on, we need the people on, and then we need to tell them what are at stake here, and what type of food do we want to have, what kind of environment do we want to have, and so on, into the discussion. I'm
aware that we're running over, and there's still a full hash of questions and things to ask. But Lawrence, maybe you'd like to sort of sum up a little bit and capture some of that.
Yeah, capture it.
Briefly, succinctly.
There you are. It's all in the chat, and look at the chat. This has been
oh, by the way, can I say before you start, we will be sending this Q&A, the links, the chat, and the video links to everybody who's here today, so don't worry about that. Now you make it.
Yeah, to pick up on Anna's last point about the challenges that we face, the climate crisis, and the last 3 times I've been in meetings in Brussels, a meeting and trying to be as difficult as it is, as sort of reasonable and laid back and pleasant as I can be, which is not that extensive really, but trying. There are people sat opposite who, as someone said in the chat earlier, that many people in policy and in the research community are decent, ethical people working in how they perceive a public interest and so on. So why are there such big differences of views, and why is it that we find such difficulty in getting an agreement or finding a consensus? I think there's 2 things. One is this issue that Sarah raised about the imminence and the urgency of biodiversity crisis, the climate crisis, and how people say we cannot afford debate. And this was people saying this to me in these meetings. We can't afford debate. We can't afford protracted participatory decision making because it takes too long. We don't have the time. And I think the answer to that is simple, but it's also complicated, which is that actually this technology, all the benefits that people claim for this technology will not work if society doesn't trust the technology, or if it's misused, if some of its potential benefits are not open, not transparent, it falls under the control of elites and corporate interests. In other words, if it's not democratically assessed and applied. Unfortunately, democracy takes time, participation takes time, traceability takes time, openness takes time. And I think the answer to that for the scientific community who are using the urgency of climate and biodiversity as a means to say, we've got to deregulate, we've got to reduce regulation, we can't have these public discussions, we've got to get on with it. Yeah, down that route lies even further erosion of democracy and citizen engagement, when actually the very nature of sustainability and equitable sustainable development is rooted in citizen engagement. So that's, guys, I think we do have to take time with this technology. On the other hand, I think this is again a part of the thing which Pat mentioned earlier, to do with policymakers, our political classes, and our scientists, which is suffering from an epidemic of hubris and a lack of humility. And that leads to an unwillingness to accept uncertainty. And accepting uncertainty, you can dress it up as risk assessment, you can dress it up as scientific methods or whatever, but ultimately it's a cultural attitude. It's knowing what we don't know, recognizing uncertainty, recognizing difficulties means being, having a degree of humility about our abilities and avoiding hubris. And that's a long-term change. So I think we've been honored, the participants here, to have 3 scientists with impeccable scientific track record who have recognized uncertainty, who have shown humility and a balance and a proportion in their comments and in their careers. And so thank you very much. As far as Beyond GM and the bigger conversation is concerned, we'll keep trying to bang on with that message and trying to make it part of the public and scientific discourse. So thank you to all participants, and thank you to you 3 presenters. It's been great. Thanks very much.
Yes, thank you. Thank you, Lars.
Lawrence.
That's fantastic.
Thank you.
Thank you all.
And with that, I think we will end. I want to also extend a thank you to our audience who have been incredibly enthusiastic and asked some really good questions. It's always painful to realize that we can't get to all of the questions, but I hope that in sending you all the links and what have you, you'll be able to carry on looking at these questions, asking them, debating them with other people, and really helping to get this message of the importance of complexity and uncertainty into the genome editing debate. So thank you very much for all of you for being here and staying here, and for your great questions. Thank you very much.